Good morning, everyone. It's very unusual for me to be the first speaker, but here I am. I have to welcome you all as well. This is a fascinating venue. I want to thank the organizing committee, the scientific committee, and the organizing committee. Really, this is, I have looked forward since 2 years ago. I was invited to this conference, and I was hoping that I will get invited again. The venue is exceptional. The app is superb, and I really encourage everyone to really get on the app. It's really magnificent. So without further delay, just a technical note. I was asked to give a lecture for 40 minutes, but now I realize it's 30, so I will adjust. Is it 30? 30, all right. Not to delay you any longer, I have an outline for what I would like to talk about today. I'm a population geneticist, but I work exclusively on complex diseases, namely type 2 diabetes and coronary artery disease. So this will be my focus. Today, I will talk on type 2 diabetes. I will start with a general classification for those of you who actually are not very familiar with type 2 diabetes quickly, and then I'll move on to the advances in the genomics of type 2 diabetes, where we are and what we've learned. And then I'll move to a small section where we talk about the architecture, if you wish, of type 2 diabetes, the genetic architecture or the genomic architecture of type 2 diabetes and the complexity of it. And then, if time permits, I will talk about my work on metabolomic, which is a follow-up to the genomics work that we've done in the past. And finally, a current project that is dear to my heart on type 2 diabetes, an NIH-awarded grant last year for us to work on specific aspects of type 2 diabetes. So, most of you know this, that diabetes is not a single phenotype. It's a spectrum of disorders and multiple pathologies with different mechanisms actually will end up leading to diabetes that you classify as a diabetes. And individuals, they do vary in their genetic background, but also other environmental insults that would lead to type 2 diabetes. And there's a lot of differences with the way individuals react to environments with a specific background. So today, diabetes is classified. We teach this to our medical students. There are many classifications for diabetes, and type 2 diabetes in particular now, there are sub-phenotypes of type 2 diabetes and sub-classification of type 2 diabetes. There are at least several subtypes of type 2 diabetes. So just to let you know that we're not talking about a single phenotype, an easily comparable phenotype, not to mention the several endophenotypes, but also within diabetes, we have the monogenic type, we have the insulin-resistant type, and what have you. Without really going through a lot of details, we know specific hallmarks of the disease, whereby type 1 diabetes occurs early on, before puberty usually, and then we have MODY. These are the most common types. Maturity onset diabetes of the young, which is a monogenic disease by large. Many loci have been identified. It's a classic monogenic form of type 2 diabetes, or diabetes if we want to be precise. And of course, what we are interested in is this type 2 diabetes, which occurs at a later stage in life. What is staggering is the numbers and the prevalence of type 2 diabetes that is really increasing throughout the world. And these figures are from the CDC, and the number of millions, this is up to 2010, you realize the importance of this disease, and we realize the importance of the environmental factors that we think are playing a major role in type 2 diabetes development. More numbers just to highlight the fact that even middle-income countries have a really staggering increase in the prevalence of type 2 diabetes, especially in earlier onset of age. So we're talking about maybe starting at about 40 to 45. The numbers have increased over the years, and they are still increasing. In particular, this number in UAE, for example, we have 17.26% between the ages of 20 to 79. I'm not sure how accurate this number is. I know for a fact that in Lebanon, if you, the recent survey that we've conducted, we have numbers much, much larger than this in a population between 40 and 60 years of age. So we know for a fact that compared to what has been done in 2001 to 2005, estimating the prevalence of diabetes in Lebanon in particular, it was in the low teens, maybe 11%. Our recent work has shown that is actually significantly more. If you go above age 55, the prevalence becomes more than 32%, which is really, really staggering. So the numbers are high, and they are likely to get higher, and this is a very scary map. We could see that in Africa and in the MENA region, we have more than 100% projection increase in the prevalence of type 2 diabetes. So this is scary everywhere in the world. It's increasing, but in particular in two spots, more than 100%, the MENA region and Africa. So how do we get diabetes? Of course, there are it's a multifactorial disease, but twin studies have consistently shown that there is a huge genetic impact combined with the environment, and this is by comparison when we do monozygotic twins versus dizygotic twins. The concordance does not drop significantly, suggesting a very high environmental factor impact. And of course, aggregation studies have also systematically and consistently demonstrated that indeed type 2 diabetes has a huge genetic component. But this genetic component is not only one gene or several genes. Actually, we are learning that it's actually much more than this, and plus many other environmental impact, and these would be within the body, but also externally as we have it. So if I were to draw really a simple cartoon of the polygenic nature of type 2 diabetes, this is something that we could actually come up with, minding that some of these variants, sorry, I'm having a hard time with the pointer here, but some of these variants, could be a bit fatter or a bit thinner, and that's what we actually are going through. So having said that, I cannot really move on to the genomics part without really talking about the Thrifty gene hypothesis, which has been developed in the 60s, saying that we are seeing most likely this increased prevalence of type 2 diabetes because as humans, we really were hunters and gatherers, and we really, our genes have been adapted so well for not having food for a long time and then eating in short bursts, and then again we really store the food. A lot of studies have been actually in support of the Thrifty hypothesis. These fasting diets that we have, the 16-hour fasting diet and on glucose metabolism in the blood. So recent studies have demonstrated that these actually have been very effective in actually regulating glucose metabolism in the blood and increased insulin activity. So perhaps there is some truth to this hypothesis. I'm a strong believer of this hypothesis, I think it's much more complex than this, but we really need to think about this. So moving on to the genetic component, a lot of GWAS studies have been conducted, and of course, initially they replicated some of the linkage analysis, candidate gene approaches, all of these things, and then they came up with a list of genes early on, the low-hanging fruits as we call them. And these early GWAS, and I participated in some of those, really they discovered a set of genes or gene variants that actually contribute to diabetes development with a certain odds ratio of varying from 1.1 to 1.4, 1.5 at the most, with multiple replications in several studies. The famous of which were the Wellcome Trust cohort, the UK type 2 diabetes study, and then we as I said, our group actually worked on this early on, and we were indeed, we replicated some of these genes. The top two SNPs that were actually in various studies worldwide, we captured those very cleanly in our population, thinking that, most of the studies that have been conducted early on were on European populations and more recently Asiatic populations. So, but really, more meta-analysis work has been conducted, and then they replicate the same. And yes, we have p-values that really are significantly low, but then the odds ratio would not increase. So what was going on here is that we were replicating the same idea, but then something that we have not figured out is where is this missing heritability? If we see concordance between monozygotic and dizygotic twins, aggregation, but this heritability has to be somewhere. And despite everything we've done so far, and I say so far, we have not reached more than maybe 10-15% of the heritability with all of these variants that we have actually captured. Well, initially we thought maybe the p-value we're putting on this, we take 10 to the minus 8 as the cutoff. Maybe we are missing some. It's true. Maybe we need a larger analysis, multiple populations, and increase the, in data analysis we say, increase the data, then we'll get a better p-value and what have you. And then perhaps we're missing these rare variants. And I really, I thought this is it. The answer is going to be in these rare variants, and that's what I've been focusing on since. So yes, and the GWAS actually had many challenges. Initially, we didn't capture all the SNPs. A lot of regions of the genomes were not actually represented. And there are more than one challenge actually with GWAS. These SNPs not necessarily coded for genes, so we had no idea how they relate to genes, the function and analysis, which tissue these variants would likely to have an impact, etc. So there are problems with these GWAS analysis that we were conducting. And then people started to refine this, and then, yes, let's pull more data in, more populations in, and let's get a way of analysis. And of course, we started seeing those very large studies conducted by many large consortia. And again, this one, an expanded genome-wide association published 2 years ago. And what is shocking is all of these numbers you could see in blue here are what has been established since the very early stages of GWAS. And look at the odds ratio. We have not really significantly increased any. And to point, the blue dots actually were the first one, the low-hanging fruits that we picked up with the highest odds ratio. So we really did not significantly improve all of this. Then last year, a year and a half ago, then a summary study came identified 143 risk variants, and again, the same idea, the same genes, with the exception of actually minor alleles. This one identified new type 2 diabetes studies, but again, very low impact, genetic impact. So basically, the summary of this very large study that I cited last, new pathways combining cardiovascular disease and type 2 diabetes and obesity and type 2 diabetes. So actually, they combined both phenotypes, saying that the same genes actually are for susceptible alleles. So these were very magnificent studies, but there was a problem that we have not really surpassed, which is, where is this heritability? So basically, a summary of what I've talked about here is we have 128 distinct plus or minus, some references for 148, what have you. But these account for 10 to 15% of the risk of type 2 diabetes. So we'll go back to that schematic that I showed you. There are multiple genes with minor effect, and it's the combination all of those, or additive effect of all of this. And of course, type 2 diabetes is a polygenic trait where both rare common variants contribute to the genetic part. So then again, after all of these studies, then let's go to the rare variants. Well, before I do that, actually, I just want to highlight the importance of actually conducting at least one GWAS or one good GWAS in populations that have not been studied before. So it's important to have all of the populations that we know of studied genetically, because you will still find some markers that actually have not been discovered before, albeit with a low impact, but these are important for us to. So there is always the importance of actually do multi-ethnic studies using GWAS and even whole genome sequencing or exome sequencing. So then there was this article which actually summarized the architecture based on all what has been done. And really, it's a fascinating piece of work where it shows that, yes, there is the importance of having, sequencing done on these families, because you will capture at least these rare variants that perhaps are important. But then when they did this, their analysis, they realized that even with the sequence analysis that was done, really not much was added to what we already know from the early GWAS. So even the genetic variants that are rare actually did not improve our results and did not significantly improve the way we are looking at type 2 diabetes. Further, they took all of the monogenic markers that have been associated with type 2 diabetes, and only a single one actually reached a genome-wide significance. So something is really amiss here. And when they modeled the architecture of this, really what was fascinating is, if you could look at the first part of this here, this is the empirical data right here, and then the model that they matched by simulation is the fact that, indeed, 75% of type 2 diabetes genetics is not rare variants, but these are the common variants that actually, in combination, they actually lead to type 2 diabetes. And let's not worry about these minor rare variants, which actually will be less than 25% component. So basically, and they say, let's not worry about what we have missed. We're not going to get any more. And I strongly believe in this. It took us a long way to get to this. And I think not only type 2 diabetes, but all complex diseases will behave in this manner. So there has to be other ways to look at type 2 diabetes. So this remaining heritability we can actually look at it from multiple perspectives. So there are structural effects. Of course, we have been looking at a one linear dimension of the genome, but the genome is much more complex. There are structural effects. There is the epistatic effect and all what the genome actually has in mind. So has in place. So the difficulty is not just by looking at single variants or copy number variants or deletions. It's actually much more complex than this. So we need other ways to look at the outcomes. One thing that really caught my attention personally is the human microbiome interaction. And I thought, well, at least it's at the tail end of the genetic expression. So can we really look at these metabolites, whether they are from the microbiome or from the actual genome? Can we look at this? And then finally, we started to experimenting with something called GWAS, which is basically looking at the epistasis factor just by rerunning what we have done in the past, but calculating or doing it taking into account the epistatic effect of genes. So moving forward, we thought, let's look at phenotypic patterns and including the metabolome with all of this. And this is what we have done over the last couple of years. And with the time remaining, I'll show you what we have really come up with. So just one slide on this. We have started experimenting with genome-wide epistasis studies, and early results are promising. But again, this is really looking at the architecture of the genome the way many others have been looking at, with a similar addition by looking at the epistatic effect and not the additive effect only. So what we have done is we said, let's cluster these phenotypes. And we've done this for cardiovascular disease, coronary artery disease, and type 2 diabetes. So basically, we have multiple phenotypes, and they cluster. And they do cluster, actually, to a certain extent. So we have, for example, in this very small, pilot, we actually clustered few individuals or few groups of individuals together. And what we've done is we said, these clusters, we have the genome, the GWAS on them, but let's do the metabolome on these individuals. And that's what we've done, basically. And this is based on data that we currently had. And we selected these phenotypes based on these clusters. And we started actually looking at the metabolites and taking these metabolites against their GWAS, as we call it, GWAS, and it becomes the mGWAS by looking at the metabolite as well. And we were lucky to discover one in particular. It's called 4-cresol. It's a metabolite that is a microbial metabolite that we discovered in the gut, which shows negative correlation with type 2 diabetes. And this is because we had really a clustered phenotype where we selected individuals who actually were more than 70 years of age with no diabetes, and our control group was people who were less than 60 years of age with type 2 diabetes. And then what we discovered is that this particular molecule had significant effect on glucose metabolism. So as you could clearly see, the plasma glucose level in time after cresol, significant decrease in glucose levels, but more importantly, had a huge effect on BMI. It also had a significant effect on insulin secretion in the body, significant increase in insulin secretion, adipose tissue. So really a wonder molecule here that we have actually triglyceride levels in the liver, all of this, and even islet cell and pancreatic cell vascularization, significant improvement, and also cell proliferation, as you could see in the lower panel here, more nuclei, and of course cells secreting insulin. So really, we couldn't have found this molecule had we not clustered these phenotypes and had we not have the GWAS data to compare for what markers we had with this particular microbiome. So the conclusion for this initial study, and it's the first phase, so this will be published, this will be online probably in a couple of weeks in Cell Reports. So basically a mechanism mediating the effect of forcresol on improved beta cell function, which involve at least now in part inhibition of the DIRCA1A gene. So do I have another 5 minutes? 5 minutes? Okay. 3 minutes. Okay, I'll be very quick. So having looked at all of these aspects of type 2 diabetes, we still believe that there is more work to be done. And recently, we came up with this idea where can we really dissect the impact of forced displacement. Unfortunately, due to the war in neighboring Syria, we had a lot of refugees in Lebanon. And we submitted a grant proposal to the NIH, was actually, they funded us by looking at the psychological stress and acute stress caused by forced displacement of refugees, perhaps with that may lead to actually increased type 2 diabetes. And just in 2 words, the idea was, can we demonstrate any impact caused by these acute perturbation in human habits on type 2 diabetes development. And we were shocked, actually. We were lucky that the refugees, the Syrian refugees in Lebanon, actually, they go to primary healthcare centers across Lebanon. So we had access to them throughout Lebanon, and then somewhat easy access whereby we can actually recruit patients from these centers. And the numbers are staggering, as you could see from this. This is based on 2018. So at least 330,000 Syrian refugees actually came for care in Lebanon. And not only they came as one time, but even as registered users. But just to sum up this, we immediately we saw a huge difference, even with hundreds of patients recruited so far, not more, is that the number of females with diabetes at an early age is staggering. It's much, much higher than we had anticipated. So what we do is we collect their HbA1c, and you could see that in females it's significantly higher. And also we have seen that the numbers and the age of women with type 2 diabetes is actually staggering. Just a few slides to show you that we've done the serum amyloid A, we've done CRP, and really it's women with much more, they have been impacted in a much more significant way than men. And this is what we actually are currently studying right now. HbA1c in females, and the trauma questionnaire. This is to actually assess the trauma, and you could see clearly in females with higher values than males. So we will continue with this, and then of course we will have the genetic background of all these patients and do some correlation studies. So without further delay, I just want to acknowledge my colleagues for this work, and I want to thank you very much for listening to me. Thank you.